Hey everyone, welcome back. In our last video, we built a dot plot algorithm completely from scratch. Today we are taking our Python skills directly into the wet lab, and this video might be a bit complicated, so I need you to stay with me throughout this video. Okay, so if you work in a biology lab, you probably do a lot of PCR to copy DNA, right? So to do PCR, you have to design short pieces of DNA called primers. So in case some of you don't know, I'll tell you PCR stands for Polymerase Chain Reaction. Okay, uh, so uh, so to do PCR, you have to design short pieces of DNA that we call primers, right? But here is the catch. You have to program your PCR matching to heat up to the exact temperature that melts the DNA double helix apart without destroying it. And this is called the melting temperature. Okay, this is called melting temperature or TM for short. Okay, so we'll write it here. TM for short. Okay, if your temperature is too high, your primers will not stick. And if it is too low, they will stick everywhere and your experiment is basically ruined. So today we are going to use BioPython to calculate these temperatures perfectly. So let's jump into VS Code directly. Right. So before computers were powerful, biologists used a quick mental math trick called the Wallace rule to estimate melting temperatures. The rule is very simple. Assign 2 degrees for every A or T and 4 degrees for every C or G. Why? Because C and G have a stronger chemical bond. Okay, so let's define a short primer sequence and calculate this manually using pure Python. Okay, so let's take a standard 20 base PCR primer. So what we will do is primer sequence is equal to what should we take? A T G C A T C G A T and G T A C. I think that should do it. Okay, and then we will perform manual Wallace rule calculation. So what we will do is count A is equal to primer sequence dot count A. Okay, we can just copy this. Okay, and what we will do here is we will make it T and this we will make C and finally this we will make G. Similarly on this side T, C and G. Okay, then what we will do is Wallace TM is equal to count is small count A plus count T multiplied by 2 and then count C plus count G multiplied by 4. Let's check all the columns whether they are correct or not. I think we'll need one column on this side and it looks correct, right? Then finally what we will do is we will print primer sequence is equal to primer sequence and print f estimated tm Wallace tm, and we can also add a small degree Celsius. Okay, so when we run this you see that we get approximately 60°C. For a quick estimate at the lab bench, this works, but it is not scientifically precise enough for complex experiment. So that's where BioPython and thermodynamics comes in. So to get a truly accurate melting temperature, we need to account for thermodynamics specifically, like how neighboring nucleotides interact with each other. So right, so a C next to a G behaves differently than a C next to an A. So doing this math by hand is a nightmare. So luckily BioPython has a module called melting temperature that handles the heavy lifting. Okay, so what we will do is we will use that library from Bio.SeqUtils import melting temp as empty. Okay, then one more from bio.sequence import sequence. Okay, then let's define my primer is equal to sequence A T G C G A T C G A T C G A T C G A T. Okay, and then uh, right, so this will convert our string to a BioPython sequence object, right? Then we will calculate TM using the Wallace rule just to check our math. Okay, so bio Wallace is equal to empty dot TM Wallace, and we will put my primer inside that, right? And finally we can print uh, BioPython Wallace TM is equal to bio Wallace, right? And we can also add degree Celsius. And if you run that, it matches our manual calculation perfectly. But now let's turn on the professional algorithms. Okay, so the next one is where the nearest neighbor algorithm is used, which is the gold standard for calculating melting temperature. Okay, so this takes into account the sequence order, salt concentration in your test tube, and DNA concentration. So we just change our function call from TM Wallace to TMNN. Okay, so what we will do is we'll call it precise TM is equal to empty dot TM, and instead of Wallace we will use TM dot NN, and similarly we will pass my primer, and finally we will print. Let's call this highly precise NN TM, and then we will use precise TM. And one more thing we have to do is dot 2f to convert it into floating point values, and we'll call it degree Celsius. And look at that difference. That the basic rule gets 60 degrees, but the advanced thermodynamics model tells us the true melting point is actually 52.26°C. If you programmed your PCR machine to 60 degrees based on the old math, your primer might not have bound properly and your experiment would have failed. So the best part about TMNN is that it allows you to simulate the exact chemical conditions of your test tube. If your lab uses a specific concentration of magnesium or sodium in your buffer, you can pass those directly into Python to adjust the math. Okay, so let's simulate something like that. What we will do is we will simulate a custom PCR buffer mix. Okay, so 50 millimolar sodium and 1.5 millimolar magnesium. Okay, so let's do that. We'll call it custom TM is equal to empty dot TM, and similarly we will use NN again, and we will use my primer sodium is equal to 50 and magnesium is equal to 1.5. Okay, then finally we will print, we will call it custom buffer TM and then we will print custom TM and close to two floating values, and then finally degree Celsius. So by passing the Na and Mg variables, BioPython instantly recalculates the complex thermodynamics tables, and you now have a custom built computational tool for your exact lab setup. And there you have it. You just wrote a script that bridges the gap between digital data and real world wet lab chemistry. And if you are working on a project where you need to design thousands of primers across an entire genome, doing this manually on a website would take weeks. But with this Python script, you can put it in a loop and calculate 10,000 melting temperatures in less than a second. The code will be available on GitHub repository as always. And if this video helped you save a PCR experiment, hit the like button, subscribe, and I will see you in the next video.